Hello everyone and welcome back to your one and only favorite channel, Let's Crack NEET UG in English. I'm Aditi Gupta, your biology mentor. And I'm live again at 5 p.m., your favorite time. And today we are going to discuss respiration in higher plants and that too about link reaction and Kreb cycle. So first let me check if I'm audible or not. And yes, I'm audible. So I welcome you all. I welcome you all to this awesome session where we are definitely going to learn the process of respiration in higher plants, and specifically we are dealing with the cellular respiration parts. So yesterday only we have started this chapter, right? And we have seen the introductory part of the respiration, the various types of respiration, the various stages of respiration, okay? And then I clearly mentioned that in this class we will be dealing with the cellular respiration process. And in yesterday's class, we have already seen the mechanism of glycolysis along with the anaerobic respiration, right? So now it's time to move ahead. And in today's class, we are going to learn about the next mechanism which takes place in case of aerobic respiration, okay? And those processes are the link reaction and the Krebs cycle, okay guys? So come on, let's begin today's session. But first, let me introduce myself and tell you something really important about Unacademy that's really going to help you in your NEET UG preparation. So guys, this is the link to our Telegram channel. You can stay connected to all of us with the help of this Telegram channel. Okay, all the upcoming lectures are updated here. Okay. Now, talking about myself, so my name is Aditi Gupta. I'm your Aditi ma'am, and I have been mentoring students for the past 6 years for their NEET UG examination. Okay, I'm here to share my knowledge so that you can, you can get the benefit of the knowledge which we are sharing here on this platform, and this will definitely help you in cracking your NEET UG examination. Now, you know that yes, the topmost team of educators are available on this amazing platform called Unacademy, and you can also be a part of the family and be under the best guidance by proceeding for Unacademy subscription. Okay, how can you proceed for Unacademy subscription? You can either visit unacademy.com or you can download Unacademy learning app from Play Store or iOS. Okay guys, first of all, if you download Unacademy learning app, you will be getting some absolutely free of cost features like the special classes and the quiz features. Okay, so each and every educator which are available on Unacademy platform has has their special classes that are absolutely free of cost. You can go and watch them. So go and explore and enhance your learning experience. That's definitely you're definitely gonna learn from this particular feature. Okay guys, talking about my timing, so I'm live daily at 11:30 a.m. mostly, right? I'm live at 11:30 a.m. on Unacademy learning app. Now talking about the subscription part, guys, so Unacademy subscription comes with two platforms, Unacademy Plus and Unacademy Iconic. And in both the platforms, you will be getting daily interactive live classes from the topmost team of educators, okay, along with a dedicated doubt clearing session. Then the complete NEET UG syllabus is covered in a structured manner, in a detailed manner, along with the PDF notes. Then your knowledge is evaluated on the basis of live tests and quizzes. And there are 50 plus educators on Unacademy subscription platform, and you will be getting access to the video lectures, to the PDF notes, and the personalized test papers of all those educators with this single subscription. So these are the benefits of Unacademy Plus. Now, if we discuss Unacademy Iconic, so all these benefits are also included here on Unacademy Iconic, but it also has some out of the box, some really amazing features. So what are those features? Each and every student here in Iconic will be getting their own personal coach who will be there to guide you in each and every step of your preparation journey. Okay, then you will be getting your own personalized study planner. Okay guys, this study planner will definitely help you so that you can utilize your time to the best of your efficiency. And these study planners will be reviewed bi-weekly and then in-depth test analysis so that you can see what mistakes you're making and what's the reason behind those mistakes. And from time to time, you will be getting expert guidance, expert motivation sessions. Okay, so this is Plus and Iconic. So in both the platforms, you will be getting India's best team of educators, interactive live classes, real classroom-like feeling. Okay, then structured courses and PDFs, live tests and quizzes. But what's new here in Iconic? Personal coach, study planner, expert guidelines, and test analysis. Okay. Now you can choose your platform according to your requirement, according to your convenience. There are multiple subscription plans available for both Plus as well as Iconic. Okay. Now, let's say if you wish to proceed for 2 year of Iconic plan, The 2 year of Iconic subscription cost you rupees 1 lakh. Okay. But if you want, if you don't want to pay the entire amount, so there is a special news for you. If you use my referral code AG001 here in this box, which is provided in the bottom, right? By applying AG001, you will be getting an instant 10% discount on your course fee. That means now the applicable fee for 2 year Iconic becomes rupees 90,000. Okay, so this is how you can get 10% discount in each and every plans which are available on Unacademy platform. Okay, guys. So that was the important information which I needed to provide you. Now, come on, let's begin with today's session. Hi, Manoj. Welcome to the session. So, guys, yesterday only we have seen that first let me revise something that yesterday only we have seen that in case of cellular respiration, the first step was the glycolytic step, right? We have seen that glycolysis consists of series of 10 reactions. Hi Avinash, welcome to the session. So as promised, I already promised you that yes, at the end of this month we will be completing the entire botany syllabus of class 11, right? And in the end of this particular class we will also be listing out the chapters which we have already finished. Okay, because this is really important. We are moving ahead, we are towards the end of the session, right? So we need to conclude. Okay, we need to conclude so that we can start the revision phase very soon. Okay, guys. So see, as I told you that, in yesterday's class, we have discussed about the glycolytic reaction. And I told you that there were 10 series of reactions, right? And in that particular step, what happened, in that particular process, what happened? One molecule of glucose, which contains 6 carbon, it was converted into 2 molecules of pyruvate or pyruvic acid. And each pyruvic acid molecule contains 3 carbon. That means one glucose molecule with 6 carbon, it breaks down into 2 pyruvic acid molecules, each with 3 carbon, right? And what were we getting? We getting 2 ATP. We were getting 2 ATP and 2 NADH2 molecules. Remember? Right? And I told you that these 2 NADH2 molecules, they will be transferring their electrons to the electron transport chain. And from there, we will be able to convert it into this energy, the energy in the electron, will be able to convert that particular energy into ATP. And one NADH2 is equivalent to how many ATP molecules? I told you it is equivalent to 3 ATP molecules. And since here 2 NADH2 molecules were produced, that means it is equivalent to 6 ATP molecules, right? So the net outcome of the glycolysis was 2 plus 6, that was 8 ATP. Then I also told you the reason that why only 2 ATP is produced in case of anaerobic respiration, although glycolysis is taking place there also, right? So we have seen till that point. And also we have seen the fate of pyruvate in case of anaerobic respiration. Okay, this pyruvate, it either gets converted into ethyl alcohol and CO2, or it gets converted into lactic acid. Okay, in the presence of enzyme lactic dehydrogenase, this pyruvate, it gets converted into lactic acid. Whereas in the presence of enzyme alcoholic dehydrogenase, it gets converted into ethyl alcohol or ethanol plus CO2. So this we have already seen in yesterday's class. Now we have to see, we have to look for the fate of pyruvate in case of aerobic respiration. So what is happening to this pyruvate when aerobic respiration is taking place? This is the thing which we need to learn in today's class. Okay. But before moving to that particular point, first you need to recall the structure of the cell. Okay, so this was the cell. Let's say this is the nucleus and this was the mitochondria. Okay, so this is the mitochondria, which contains finger-like folds. This is the cristae. Inner membrane of mitochondria is what? Inner membrane of mitochondria is cristae. And now the glycolysis was taking place here in the cytoplasm, and from the glycolysis, what was formed? Pyruvic acid was formed, right? Now, this pyruvic acid, my dear students, is transported actively to the matrix region of the mitochondria. This is what? This is the matrix of mitochondria. This is the matrix of mitochondria. Okay. So now what happens to this pyruvate in case of aerobic respiration? So what happens? This pyruvic acid, it is actively transported to the matrix of the mitochondria. See, that means some amount of ATP is consumed at this step. Okay, and then what happens? Here in this matrix region, 2 cycles are going to take place. First one is the link reaction. First one is what? First one is the link reaction. Okay, and through the link reaction, acetyl coenzyme A will be formed. and that acetyl coenzyme A, which is formed with the help of the link reaction, will be participating in the Krebs cycle, which is also taking place in the mitochondrial matrix. So that means in today's class we are going to focus on the reaction which are taking place in the mitochondrial matrix, the link reaction and the Krebs cycle. Okay guys, so come on, let's begin with the link reaction. This link reaction, it is also known as the gateway reaction. It is also known as what? It is also known as the gateway reaction. Okay, basically it is a connecting link. Okay, it is a connecting link between the glycolysis and the Krebs cycle, and that is why the term link or the gateway reaction is given to this particular process. Okay, so it is a connecting link between the glycolytic process, that means the glycolysis, and Krebs cycle. Okay, we know that the pyruvic acid which is generated in the case of glycolysis, it gets converted into acetyl coenzyme A in this link reaction. Okay, and that acetyl coenzyme A then enters into the Krebs cycle. Okay guys, so what is now actually happening in this link reaction? So I told you that, you know, with one molecule of glucose, two molecules of pyruvic acid are formed. So two molecules of this pyruvic acid or pyruvate is formed. Okay, although we don't need to remember the formula, I'm simply writing it. Okay, so this pyruvic acid or pyruvate, it reacts with coenzyme A molecule, and in the presence of enzyme pyruvate dehydrogenase complex, in the presence of which enzyme? Pyruvate dehydrogenase complex. And when I say it is a complex, that means it consists of a series of enzymes. Okay, so this pyruvate dehydrogenase complex is a series of enzymes. It contains enzyme dehydrogenase for the process of oxidation. Okay, for the process of oxidation. Then the enzyme decarboxylase for the process of decarboxylation. Then the enzyme transacetylase for the formation of this bond between the coenzyme A and the pyruvate. Okay, so these are some of the enzymes, although there are more enzymes which are present in this complex, but I am mentioning about these three. These are the three important enzymes: the dehydrogenase, the decarboxylase, and the transacetylase. Okay, these are the three major enzymes found in this particular complex. And now in the presence of this enzyme complex, this pyruvate molecule, it gets converted into two molecules of acetyl coenzyme A. It gets converted into acetyl coenzyme A. This is what? This is acetyl coenzyme A. And for the first time in the history of respiration, okay, for the first time here, the CO2 is released. Remember, students who have attended yesterday's class, they know that in the process of glycolysis, neither the oxygen was getting consumed and nor the CO2 was produced. Okay, so oxygen was also not consumed and CO2 was also not produced, but this is the first reaction in case of respiration where the CO2 molecule is getting produced. Okay, that means the process of decarboxylation is taking place, and also oxidation is taking place. And oxidation, when oxidation takes place, what happens? For example, this pyruvate, it is oxidized to acetyl coenzyme A. Okay, that means it has lost its electron, its hydrogen. So we need an electron hydrogen carrier, and here two molecules of NAD+ are present, which will receive those electron hydrogen and they will convert into two molecules of NADH2. Why two molecules? Because two molecules of pyruvate are present, right, from one molecule of glucose, and that is why two molecules of reducing electron hydrogen carrier is present over here. Okay guys, one more important thing. This enzyme complex, pyruvate dehydrogenase complex, this enzyme requires 5 must or mandatory components. What are those components? It is written over here. They need magnesium ion. They need what? Magnesium ion. Then lipoic acid. La stands for lipoic acid. TPP stands for thymine pyrophosphate. Vitamin B derivative, right? Thymine pyrophosphate. Okay, then NAD is nicotinamide adenine dinucleotide. Then nicotinamide NAD molecule, it is again a vitamin B derivative, right, derived from niacin. Then coenzyme A. Okay, all these 5 components are mandatory for this reaction to take place, and this reaction is taking place in the mitochondrial matrix. And what exactly is happening in this reaction? Oxidative decarboxylation is happening in this reaction. Why are we calling it as decarboxylation? Because CO2 is getting released. From each pyruvic acid molecule, one carbon is released as carbon dioxide. Okay, so from 2 pyruvic acid molecule, 2 carbon dioxide are released. And oxidation is also taking place at the same time. That is why we are using the term oxidative decarboxylation is taking place. Okay, guys. So I hope this process is clear. And this is the link reaction. Why? Because the acetyl coenzyme A, which is produced over here. Now this acetyl coenzyme A will be entering into the Kreb cycle. Okay, it will be entering into the Kreb cycle. That means the next stage of the cellular respiration, and then it will ultimately break into carbon dioxide and water molecule. Okay, and water molecule will be actually produced as a metabolic water. So this acetyl coenzyme is the connecting link which is produced as a result of the link reaction, and that is why this link reaction acts as a link between the glycolysis and the Krebs cycle. I hope this point is clear. Okay, guys. Now moving ahead. And let us have a clear understanding of the processes which are taking place in case of the next step, which is the Krebs cycle. Okay, this Krebs cycle, it is named Kreb because it was discovered by H.A. Kreb, right? It is also known as TCA cycle. What is this TCA cycle? Basically, it is tricarboxylic acid cycle. TCA is tricarboxylic acid cycle. Okay. So what is actually happening and where is this process taking place? So see, first of all, this Krebs cycle, it is also taking place in the mitochondrial matrix. Where? In the mitochondrial matrix. Okay, that means all the enzymes which are involved in the process of Krebs cycle, they are present in the matrix of mitochondria, but there is one special enzyme which is also known as marker enzyme. Which enzyme? Marker enzyme. And this special enzyme is succinate dehydrogenase. Okay, and this enzyme, my dear student, is found in the inner membrane of mitochondria. It is not present in the mitochondrial matrix. Okay, it is found in the inner membrane of the mitochondria. Okay, inner membrane of mitochondria. And when you will study the electron transport chain, there you will see that yes, it is one of the component of the electron transport chain. Okay, we know that the electron transport chain here in mitochondria is present in the inner mitochondrial membrane, right? And the succinate dehydrogenase enzyme, it is one of the components. Okay, so it is found in the inner membrane of mitochondria, and rest all the enzymes are found in the matrix of the mitochondria. Okay, now what happens, basically what is happening? So first of all, oxidation is taking place, because we know respiration is a process of breakdown. Okay, so the oxidation is taking place, breakdown of the substance is taking place, and acetyl coenzyme is entering into the Krebs cycle, and it is received by a 4 carbon molecule, which is oxaloacetic acid. Okay, so this oxaloacetic acid, this is a 4 carbon organic acid. Okay, and this is the first molecule, or we can say the acceptor molecule, which is going to accept the acetyl coenzyme A. Okay, which is getting produced in the link reaction, and that too is taking place in the mitochondrial matrix. Okay, and the cycles start. Okay, so let us move ahead and have a look on the various steps of the cycle, of the Krebs cycle basically. Okay, so see what's happening. This is oxaloacetic acid. Okay, now acetyl coenzyme A is entering. Okay, first of all, we will be considering the cycle considering one acetyl coenzyme A molecule. But we have to keep this point in mind that from one glucose molecule, 2 pyruvic acid are formed, and from those 2 pyruvic acid, 2 acetyl coenzyme will be formed. That means from one glucose, how many acetyl coenzymes are there? 2 acetyl coenzyme A are there, and plus 2 CO2, which we have already received during the process of link reaction. Okay, during the process of link reaction. Now, in order for complete oxidation of one glucose molecule, we need to run this Krebs cycle twice, because there are 2 acetyl coenzyme A molecules. Okay, guys. Now see what happens. This oxaloacetic acid is a 4 carbon compound, and this acetyl coenzyme A contains 2 carbons. Okay, and here an enzyme which is known as citrate synthase. Okay, citrate synthase is present, and a simple condensation reaction will take place. And what happens? A 6 carbon intermediate, which is citrate or citric acid, is formed. Remember, I keep on repeating this point that the acids inside the body, they are present in their salt form, and that is why we are using the suffix ate every time. Okay, be it pyruvate, be it oxaloacetate, be it citrate. Okay, I'm not writing the term citric acid, although we can use it. Okay, so this citrate, it is a 6-carbon molecule. It contains 3 carboxyl group, and that is why it is known as tricarboxylic acid. So this is the first form stable product in case of Krebs cycle, and that is why it is also known as the TCA cycle. Because this is the same fact we have seen in the process of photosynthesis also. What was happening in case of C3 cycle? The first stable product was a 3-carbon compound PGA. In C4 cycle, the first stable product was a 4-carbon compound. Okay. Here the TCA cycle, because the first stable product is a citrate. Okay. Now what happens? This citrate, it gets converted into its isomeric form, which is isocitrate. In the presence of enzyme aconitase, which needs parasite. Okay. So this isocitrate. Now this isocitrate is a 6-carbon compound. Now this isocitrate, it undergoes the process of oxidation. Remember, in the Krebs cycle, there are 4 sites of oxidation. Now this is the first site of oxidation. Now here what will happen, since the oxidation is taking place, so the enzyme dehydrogenase will be present. Okay, the enzyme dehydrogenase will be present, and electron hydrogen acceptor will also be there. Electron hydrogen acceptor molecule is also there. Okay, and now this isocitrate, upon oxidation, it converts into oxalosuccinate. Okay, but this oxalosuccinate, it is also a 6-carbon compound. It is very, very, very unstable. So as soon as it forms, it is further broken down. Okay, so basically we can say that it is not even formed, because it is so damn unstable that it is continuously breaking down. Okay, so this isocitrate, upon oxidation, it for a temporary moment it forms oxalosuccinate, but this oxalosuccinate, it undergoes the process of decarboxylation. Okay, and it releases a CO2 molecule here, and it forms alpha ketoglutarate. It forms what? Alpha ketoglutarate. Okay, guys. Hi, Durga. Welcome to the session. So I hope you guys are enjoying the session and each and every concepts are getting clear. Okay. Do let me know if you have any doubts, guys. Okay. See, since the process of decarboxylation is taking place, the enzyme involved here is decarboxylase. Okay. And this enzyme requires manganese ion as a cofactor. Okay. Now, this alpha ketoglutarate is a 5-carbon compound. It is a 5-carbon compound. Now this 5-carbon compound, it undergoes the process of oxidative decarboxylation. That means CO2 will be released once again, and the process of oxidation is taking place. That means electron hydrogen carrier is present, and that electron hydrogen carrier again is NAD molecule. And the enzyme which is involved here is dehydrogenase complex. It is dehydrogenase complex. Okay. This enzyme also requires, lipoic acid, thymine pyrophosphate, magnesium ion, coenzyme A. All these components are also required here in this particular case also. Okay, and what happens? This alpha ketoglutarate, a 5-carbon compound, it finally converts into succinyl coenzyme A. Which is a 4-carbon compound. Which is what? Which is a 4-carbon compound. I hope this point is visible to you. Okay, so what has happened till now? With the help of the link reaction, pyruvic acid molecule is converted into acetyl coenzyme A molecule. Pyruvic acid contains 3 carbon. Acetyl coenzyme A contains 2 carbon. So 1 carbon is released in this process as carbon dioxide, and this was the first reaction where we have seen the formation or the release of carbon as carbon dioxide, and that reaction was the link reaction. And this question has been asked multiple times in the previous year medical examination. So pay special attention to this point. Okay, now this acetyl coenzyme A has entered into the Krebs cycle, and now we are discussing the Krebs cycle only. Okay, so what is happening? The oxaloacetic acid, which is a 4-carbon compound, it is saying hello to the acetyl coenzyme A molecule, right? Because when a guest enters or when a guest comes to your house, what happens? You open the door and you greet them, right? So hello and hi, and then you ask them to come inside and sit. Okay, So basically, this acetyl coenzyme A is also a guest molecule. And oxaloacetic acid, it is basically, it is the host and it is accepting, it is saying hello, it is greeting the acetyl coenzyme A molecule. And this is how, a combination, a bond is developed, right? So with the help of the condensation reaction, citric acid is formed. This is the first formed product, which contains 3 carboxyl group, and that is why tricarboxylic acid name is given to this particular compound. Okay. Now this citric acid, it gets converted into its isomeric form, which is isocitrate, again a 6-carbon compound. This isocitrate, it undergoes the process of oxidation in the presence of NAD molecule, because NAD will be acting as hydrogen electron carrier. Okay. And the enzyme is dehydrogenase. This dehydrogenase is very famous whenever we discuss about the redox reaction. See, when I am saying that the isocitrate is getting oxidized, at the same time this NAD molecule is getting reduced now, because it is accepting the electron and hydrogen, which is being released from the isocitrate molecule. That means both oxidation and reduction reaction are taking place at the same time. That means this is a redox reaction, and dehydrogenase enzyme is catalyzing the redox reaction. Okay. Now what happens? A highly unstable intermediate, which is a 6-carbon oxalosuccinate, is formed transiently only, but soon as it is formed, it undergoes degradation again in the presence of enzyme decarboxylase. What happens this time? This time a carbon dioxide molecule is released again. Okay, so count the carbon number. Acetyl coenzyme A contains 2 carbon. Out of the 2 carbon, 1 we have received here, right? Now it forms a 5-carbon intermediate, which is alpha ketoglutarate. Okay. Now this alpha ketoglutarate, in the presence of enzyme dehydrogenase complex, okay, which also requires magnesium ion, lipoic acid, thymine pyrophosphate, NAD as electron hydrogen carrier, and coenzyme A. All these components are needed here for this enzyme complex also, and it gets converted into a 4-carbon intermediate, which is succinyl coenzyme A. Okay. Now what happens? This succinyl coenzyme A, that means the bond between the succinyl group and the coenzyme A, this bond is a high energy bond. Okay, so what happens in the presence of enzyme thiokinase? In the presence of enzyme thiokinase, this bond between the succinyl and coenzyme A is broken. That means the coenzyme A molecule is released. Okay, and the succinyl coenzyme A gets converted into succinic acid. It gets converted into succinic acid. I'll be writing succinate only. Okay, I'll be writing succinate to save the space. Okay. Now one more important thing has taken place here. What is happening? The succinyl coenzyme A, I told you that the bond between the succinyl and the coenzyme A is a high energy bond. Okay. In the presence of enzyme thiokinase, the bond is broken and succinyl coenzyme A is getting converted into succinate, which is again a 4-carbon compound. But the energy released in this process, what happens? The energy released in this process will be utilized in the formation of a ATP molecule. In the formation of a ATP molecule. So what happens here? GDP, it is similar to ATP molecule. It forms GDP combines with inorganic phosphate, and it forms one molecule of GTP. Okay. So this is again substrate level phosphorylation. This is again what? This is again a substrate level phosphorylation, because the substrate itself is providing energy for the formation of the bond. Okay, so this is substrate level phosphorylation. We have seen two reaction in case of glycolysis. There also we saw that the formation of ATP was because of the substrate level phosphorylation, right? Here, this is the third step where substrate level phosphorylation is taking place. Okay, although in the end we will also be summarizing them, so do not worry. Now this succinic acid or succinate, it gets converted into the fumaric acid or fumarate. This reaction is again special. This reaction is special, and let me change the color of the marker for this particular reaction. See what happens. Why is this reaction special? Because this reaction is being catalyzed by the enzyme succinic dehydrogenase. So I told you that all the enzyme of the Kreb cycle are present in the mitochondrial matrix except this succinic dehydrogenase, which is acting or behaving as a marker enzyme for the mitochondria. And the electron hydrogen carrier associated with this particular dehydrogenase is not NAD, but it is FAD. Okay, so see, wherever the dehydrogenase enzyme was present, the dehydrogenase has their own electron hydrogen carrier associated with them. In both these steps, here and here, you can see that NAD molecule is acting as electron hydrogen carrier. But here in this case, in case of succinic dehydrogenase, it is having FAD molecule, flavin adenine dinucleotide, as electron hydrogen carrier. Okay, so now this FAD, it gets converted into FADH2 because the process of oxidation has taken place. So this is the third site of oxidation. Thiokinase and succinic dehydrogenase, these are two different enzymes. Do not get confused. Thiokinase catalyzed the reaction in the previous step where the succinyl coenzyme A was getting converted into the succinate molecule. Now the succinic acid or succinate is getting converted into fumarate in the presence of enzyme succinic dehydrogenase, and the electron hydrogen carrier here is FAD, and it is getting converted into FADH2 molecule. Okay, so this is the important reaction because oxidation is taking place, and here comes the important marker enzyme, and also the electron hydrogen carrier here is not NAD molecule, here it is FAD molecule. So question is asked, question has been asked multiple times from this particular reaction. Okay, that is why this is a special reaction. Now what happens? Let me again change the color. This fumarate, it gets converted into malic acid or malate in the presence of enzyme fumarase. I'm not writing the name of the enzyme to save the space. And this malate, it is further oxidized to oxaloacetic acid in the presence of enzyme dehydrogenase. Okay, and here also the electron hydrogen acceptor is NAD molecule, and it gets converted into NADH2. It gets converted into NADH2. So this is how the Krebs cycle is taking place in the matrix of the mitochondria, and let us see that what are those sites where the process of oxidation has taken place. So this is the first reaction where the oxidation has taken place. Okay. Now this is the second reaction where the oxidation has taken place. This is the third reaction where the oxidation has taken place, and this is the fourth reaction where oxidation has taken place. So that means oxidation is taking place total for the 4 times. Right? Now, out of the 4 oxidation reaction which has taken place, three reactions. In the three reactions has NAD as electron hydrogen carrier, right? This first reaction, second reaction, and the last reaction, right? So let us identify those spots where NAD is acting as an electron hydrogen carrier. See, when the oxidation is taking place at the last 6 carbon compound, which is the isocitrate, here what is happening? The electron hydrogen carrier is NAD molecule. Okay, and here the process of oxidation is taking place at the last 6 carbon. When the first 4 carbon intermediate is formed, that means the succinyl coenzyme is getting formed. There also, in that process also, oxidation is taking place, and NAD is acting as a electron hydrogen acceptor. Okay, so the second site of oxidation is when first intermediate of 4 carbon compound is formed, and the last oxidation reaction where NAD is acting as electron hydrogen acceptor and where the oxidation itself is taking place is the reaction where the last 4 carbon compound is formed. So you have to remember the 644. 644 is what? This is the last 6 carbon compound. This 4 is what? First 4 carbon compound, and this 4 is what? Last 4 carbon compound. Or in all these 3 sites, what has happened? Oxidation has taken place, and NAD is acting as electron hydrogen carrier. Okay, and this reaction, my dear students, this reaction where the oxidation has taken place in between succinate and fumarate in the presence of enzyme succinic dehydrogenase, it itself is a very famous term. Okay, it itself is a very famous reaction. So you won't find any difficulty in remembering this particular reaction. Okay, and remember, with this succinic dehydrogenase, FAD is always associated. So with the FAD is always associated. Okay, so with succinic dehydrogenase, this FAD is always associated. That means in this reaction, electron hydrogen carrier is FAD molecule. I hope this point is clear. Is this point clear? Okay, so now in questions, in examination, sometimes the sequence is asked. Okay, the sequence is asked. Now pay attention to this molecule, alpha ketoglutarate. Okay, this is involved in in protein biosynthesis. Okay, although I'll be covering this point again. So see, how can we remember all these intermediates? So there is a simple mnemonic which is easily available on the internet. So I'm writing it over here. See, can I keep selling sweet for money officer. Okay, so can I keep selling sweet for money officer? This is the simple mnemonic which is easily available on the internet. Okay, I have not developed this mnemonic basically. Generally, I do not prefer to design a mnemonic. Okay, I usually take it from the internet because when it is easily available, why to waste time, right? So can I keep selling sweet for money officer? So this is the mnemonic through which you will be able to recall all the reactions, okay, all the reactions which are taking place in the Krebs cycle. I hope this point is clear. Okay, now let us go through the points which are mentioned over here. So in the Krebs cycle, as I told you, that all the enzymes except succinic dehydrogenase are present in the matrix of mitochondria. Okay. Now oxaloacetic acid is the acceptor molecule and citric acid. Okay. This is tricarboxylic acid, which is the first formed product. Now oxidation takes at 4 sites. Now what are the 4 sites? I told you the trick. The 4 sites are, you need to recall the 644. Okay. What is this 644? Last 6 carbon compound, which was the isocitrate. Okay. Then the first 4 carbon compound, which was the succinyl coenzyme A. Okay. And in the last 4 carbon compound, which was the oxaloacetate. Okay. So the oxidation was taking place here also, and oxidation was taking place in the formation of succinyl coenzyme A also from the alpha ketoglutarate, and oxidation was taking place here also in the formation of oxaloacetate from the malic acid, right? And in all the 3 cases, electron hydrogen acceptor is NAD molecule, and it is converting into NADH2, right? So these are the 3 sides of oxidation. Now what was the another side of oxidation? Between succinate to fumarate. Okay, and this is a very famous example. You need to remember this reaction, and this reaction is catalyzed by enzyme succinic dehydrogenase, which is not present in the mitochondrial matrix. Okay, so this is succinic dehydrogenase, and it has FAD molecule as electron hydrogen acceptor. It has FAD molecule as electron hydrogen acceptor. Okay, so this is the 4 sites of oxidation. I hope now your problem is solved. Okay, now let us see what is produced. Okay, now let us see the energetics of the Krebs cycle. So I told you, see, this is the Krebs cycle. Okay, a clearly written Krebs cycle. Let us go through it first. See, this is oxaloacetic acid or oxaloacetate. Acetyl coenzyme is accepted by the oxaloacetate, and in the presence of enzyme citrate synthase, citrate is formed. Now this citrate in the presence of aconitase converts into isocitrate. And see here the first, both the steps are merged here. Here oxidative decarboxylation is taking place. That means oxidation as well as decarboxylation, both processes taking place at the same time. See, CO2 is released, that means decarboxylation is taking place, and oxidation is also taking place at the same time, and electron hydrogen carrier here is NAD molecule. Okay. Now what is formed? Alpha ketoglutarate is formed, which is a 5 carbon intermediate. Okay, this alpha ketoglutarate, when it converts into succinyl coenzyme A, a 4 carbon compound, again decarboxylation is taking place, and not only decarboxylation, it is again oxidative decarboxylation. Oxidation is also taking place, and NAD is acting as electron hydrogen carrier, and C, the CO2 is released. So acetyl coenzyme A, it consists of 2 carbons, and both the carbons are released in the form of CO2 molecules here only. But then why is the cycle running? Why is it continuing ahead also? It is continuing in order to regenerate the oxaloacetate molecule, because remember, this is a cycle, and in order for the cycle to continue, we must have this, this receptor molecule. Otherwise, when the next time the acetyl coenzyme A will be entering, what molecule will be there to accept it? So in order to regenerate the oxaloacetate, this cycle is continuing further. Okay. Now this succinyl coenzyme A, the bond between the succinyl and the coenzyme A is a high energy bond, and it is broken by the thiokinase enzyme, and it converts into succinate. But here the energy is released, and that energy is utilized in the formation of a GTP molecule. Okay. And this, my dear student, is again substrate level phosphorylation. What is substrate level phosphorylation? When the substrate release energy for the process of ATP formation without the involvement of electron transport chain. So this is substrate level phosphorylation. Now what happens? See, this, let me change the color. I think red is not visible over this pink color. See what happens. This GTP, it combines with ADP at the same time, and it forms ATP, and the GDP converts into GTP. Okay, so this is the reaction. That is why every time, when I'm saying a GTP is formed, so I'm anyways counting it as ATP molecule because this reaction is taking place parallelly. Okay, so that means one ATP molecule is formed here in this step when the succinyl coenzyme A is getting converted into the succinate molecule. Now here in the presence of the marker enzyme succinic dehydrogenase, okay, succinic dehydrogenase, the succinate is getting oxidized into fumarate and the electron hydrogen carrier is fat. This is the important point. The question is asked from this point. Okay, now this fumarate, it gets converted into malate in the presence of enzyme fumarase. And the final oxidation takes place here in the presence of enzyme dehydrogenase. Again, the electron hydrogen carrier is NAD molecule. Okay, so the 4 site of oxidation is the last 6 carbon compound, C, the first 4 carbon compound formation, that means this one, the last 4 carbon compound. These are the 3 site of oxidation and 1 site of oxidation, we anyways remember. Okay, now I was talking about the energetics. What are the outcomes of the Kreb cycle? So let us pay attention to it. Okay. So with each Kreb cycle, that means for each molecule of acetyl coenzyme A, per acetyl coenzyme A molecule, what are we getting? We are getting 3 NADH2 molecule, 1 FADH2 molecule and 1 ATP. Right? Am I correct? Because we are getting 3 NADH2 at 3 sites of oxidation, 1 FADH2 where 1, at another site of oxidation, and 1 ATP at this particular process by the process of substrate level phosphorylation. So this question may be asked that the process of ATP formation in the Kreb cycle is through which one of the following process? Oxidative phosphorylation, photophosphorylation or substrate level phosphorylation. So it is because of substrate level phosphorylation. Yes. What are the other 2 reactions which we have seen for substrate level phosphorylation? Remember substrate level phosphorylation in the process of glycolysis when 1,3-bi-PGA was getting converted into 3-PGA. Remember, right? So it was the time when the substrate level phosphorylation was taking place, and another time was when the phosphoenol pyruvate was getting converted into pyruvic acid. In both these reactions, substrate level phosphorylation was taking place and ATP was produced. Right? And the third reaction is this one. When the succinyl coenzyme A is getting converted into succinate. So these are the 3 reactions in the process of respiration where substrate level phosphorylation is taking place. Okay. Now see, as I told you that one glucose molecule, it will break into 2 acetyl coenzyme A molecule. Right? So that means, to metabolize one glucose molecule, the Kreb cycle needs to run twice because per glucose molecule, 2 acetyl coenzyme A are produced, right? That means, for one glucose molecule, per glucose molecule, how many times the Kreb cycle is running? 2 times. Okay, 2 times the Kreb cycle is running. So that means, what are the outcomes? Per glucose molecule, 6 NADH2 molecule, right? 2 FADH2 molecule and 2 ATPs are produced per glucose molecule in a Kreb cycle. And we know that each NADH2 molecule, let me take the screen. Okay, so per glucose molecule I told, right? For one glucose in Kreb cycle, what are we getting? We are getting 6 NADH2 molecule, 2 FADH2 molecule and 2 ATP. And these ATPs are through substrate level phosphorylation. Okay. Now, as I told you that 1 NADH2 molecule is equivalent to 3 ATP. So 6 into 3 it becomes 18 ATPs. Okay. 1 FADH2 molecule is equivalent to 2 ATP. So 2 into 2 it becomes 4 ATP. Right. And here we have 2 ATPs direct. But we know that these molecules, they will be delivering their electron to the electron transport chain. And then only the process of ATP formation takes place. Okay. So basically these are not the direct form of energy which we can use. Okay. They are the indirect form of energy which will be getting converted into ATP molecule once they will be transferring their electron to the electron transport chain. Okay. So, but in order to calculate the total number of ATP produced from the Kreb cycle, so it becomes 18 plus 4. Okay. Plus 2. That means plus 2. I'm writing it over here. So that means it becomes 24 ATP molecules. Becomes how many ATP molecules? 24 ATP molecules. That means per molecule of acetyl coenzyme A, in a Kreb cycle, 12 ATPs are getting produced. In a Kreb cycle, 12 ATPs are getting produced. On the other hand, per molecule of pyruvic acid, Per molecule of pyruvic acid, how many ATPs are getting produced? In this case, in order to calculate it, you need to include the NADH2 which is produced in the link reaction. Remember, in the link reaction, from one molecule of pyruvate, one molecule of acetyl coenzyme A, and one CO2 was formed, and one NADH2 was produced, right? So per pyruvate, that means one NADH2 is included more, and one NADH2 is equivalent to how many ATPs? 3 ATPs. Okay, this is one NADH2. That means 3 ATPs more. That means now it becomes 12 plus 3. So per pyruvic acid molecule, it yields 15 ATP. Per pyruvic acid molecule is yielding how many ATPs? 15 ATP. Okay, guys? Okay. So what do we have till now? In the glycolysis, what we have received? In the glycolysis, I told you we had 2 ATPs, direct ATP from the substrate level phosphorylation, and 2 NADH2 molecules, right? Which is equivalent to 6 ATP, which is equivalent to 6 ATP. That means the outcome of glycolysis was 8 ATP. Now what you have to do? You have to summarize each and everything which you are receiving in order to get the total number of ATP produced in case of respiration. Tell me, guys, is this point clear? Because most of you, yes, RK12 नहीं आया समझ में. So this see, yes, what is happening? First of all, let me take another screen. This was glycolysis. Okay, this was the link reaction, and this is the Kreb cycle. So we have studied till this point, right? Any confusion till this point? No. What was happening in glycolysis? One glucose molecule, it got converted into 2 molecules of pyruvate, and as a result, 2 ATPs and 2 NADH2 molecules were produced. Okay, these are the outcomes of the glycolysis. Now in the link reaction, 2 molecules of pyruvate, it got converted into 2 molecules of acetyl coenzyme A, and in this process 2 NADH2 molecules were produced. 2 NADH2 molecules was produced. Okay, this is 2 NADH2 molecules, right? Now in the Kreb cycle, 2 acetyl coenzyme A, it is getting converted into 2 CO2 molecules. Okay, and in this process we are getting 6 NADH2 molecule, 2 FADH2 molecule, and 1 ATP molecule, right? So now what is the total energetics of respiration? Energetics of respiration. Okay, although we know very well that the NADH2 and the FADH2 molecules, they do have energy stored in them, but still the ATP formation has not taken place. The ATP formation from these energy which are stored in the NADH2 and FADH2 molecule will be taking place in the ETS process, or electron transport chain process, right? But we know that each NADH2 molecule is equivalent to 3 ATP, and each FADH2 molecule is equivalent to 2 ATP. Okay, so now let us count the total number of ATPs which are directly produced. So it is 2 plus 1 here. Okay, basically it will be 2 ATP. So basically it is 2 plus 2 here. So direct ATP which is produced with the help of substrate level phosphorylation are 4 ATPs. Okay, now how many NADH2s does we have? So 2 plus 2, 4, and plus 6, that means 10 NADH2. Okay, so 10 into 3, that means 30 ATPs, because each NADH2 molecule is equivalent to 3 ATP, right? Now how many FADH2s does we have? We have 2 FADH2, and each FADH2 is equivalent to 2 ATP. That means 2 into 2 is become 4. So now what is the total number of ATP which are getting produced? 30 plus 4 plus 4. That means total 38 ATPs are produced in the case of respiration. Okay, from one glucose molecule, total 38 ATPs are produced in case of respiration. Okay, normally in general. Although in the reaction in our first class we have seen that we have also written 36/38 ATP. Remember? What's the logic behind this 36 ATP? That I will be explaining you when we will be studying the electron transport chain. Okay, but for now you need to understand this concept, that how this 38 ATPs are produced per glucose molecule. Now I told you that one molecule of pyruvic acid, how many ATPs will it be producing? So one molecule of pyruvic acid means one NADH2 and all these things. Okay, that means it was equivalent to 15 ATP. One acetyl coenzyme is equivalent to 12 ATP. So we have seen all the calculation. I hope now this point is clear. Is this point clear now? So this is the energetics of the Krebs cycle, and this is how 38 ATPs are produced in the process of respiration. Now, there comes another important point, and that point is what? See, although we know that in case of respiration, the process of catabolism is taking place, that means breakdown of glucose is taking place, or we can say breakdown of substrate is taking place in order to release energy. So respiration is a catabolic pathway. But respiration is referred to as amphibolic pathway. And why is it so? So in order to understand this, first you need to understand that, we have started studying the cycle starting from the glucose. But what happens if the fats want to enter into the cycle? That means the breakdown of fats needs to take place. Or what happens when the protein breakdown takes place? How will they enter into the cycle? So what is the process? See, the fat molecule, they break down into fatty acid and glycerol molecule. This glycerol molecule, it gets converted into PGAL or dihydroxyacetone phosphate, that means DHAP. Okay, and then through PGAL or DHAP, they enter into the cycle. That means, the PGAL is involved in the glycolysis pathway. That means this glycerol, it enter into the respiration pathway through the glycolytic pathway. This fatty acid, if it needs to enter into the pathway of respiration, it first gets converted into acetyl coenzyme A by the process of beta oxidation, and then this acetyl coenzyme A, it enters into the Krebs cycle. Okay, talking about the proteins. So the proteins, they are converted into various amino acids. Okay, and they can enter into the respiratory pathway, either converting into the pyruvic acid. Okay, or they can also enter in the form of into the Krebs cycle directly, because the alpha ketoglutarate, the 5-carbon intermediate which was produced in the Krebs cycle, it is an important connecting link between the biosynthetic pathway, okay, of proteins. Various amino acids are produced with the help of this intermediate. Okay, so this is how different compounds enter into the cycle. Okay, now as I told you that yes, respiration is an amphibolic pathway. So let's see how the different intermediates of respiration are involved in the biosynthesis of other molecules. For example, the phosphoglyceraldehyde molecule produced in case of glycolysis, it gets converted into glycerol whenever the formation of fatty acid or lipids is going to take place. So see, the phosphoglyceraldehyde is withdrawn from the cycle when fat synthesis is required. So it is involved in the synthetic phase, that means anabolic reaction. Similarly, the phosphoglycerate, this PGA, it is withdrawn from the glycolytic pathway whenever the synthesis of amino acid serine, glycine, or cysteine has to take place. That means PGA is an intermediate or is a precursor for these amino acids to synthesize. Another an important example of anabolic pathway, right? The pyruvate which is formed as an end product of glycolysis, it is involved or utilized in the production of alanine, another type of amino acid. Okay, so the pyruvate, it is involved in the formation of alanine. Okay, now let's say alanine needs to enter into the Krebs cycle. So what will it do? Or in the respiratory cycle, what will it do? The alanine will get converted into the pyruvate. Okay, serine, cysteine, glycine, they can convert back into the PGA molecule and then enter into the cycle. This is how they enter, they interconvert and they enter into the cycle, right? Then the acetyl coenzyme A. This is a really important molecule because this is a connecting link between the metabolic pathway of carbohydrates, proteins, and fats. Okay, this acetyl coenzyme A, it is involved in the synthesis of, I told you, fatty acids. See, fatty acid is synthesized by this acetyl coenzyme A. Okay, similarly, the intermediate succinyl coenzyme A, remember the intermediate succinyl coenzyme A, which was formed in the Krebs cycle, it is involved in the formation of a chlorophyll molecule. Okay, the cytochrome molecule, which is involved in the electron transport chain. Then the phytochrome molecule. Okay, so again it is involved in the biosynthetic pathway. Okay, similarly the oxaloacetic acid and the alpha ketoglutarate, Okay, oxaloacetic acid and alpha ketoglutarate, they are involved in the synthesis of various amino acids. They're involved in the synthesis of various amino acids. Okay, so this is how the respiratory pathway is connected to various other synthetic pathway or synthesis pathway. Okay, and that is why we call respiration is an amphibolic pathway. Okay, because the intermediates of respiratory pathway are involved in the anabolic reactions. And what is amphibolic? When the catabolic and the anabolic reaction, both type of reactions are taking place. This is amphibolic, right? So this is the reason why respiration is termed as an amphibolic pathway. I hope this point is clear. Okay, so I have told you like the point from where the fats and the proteins will be entering into the cycle, the intermediates which are involved in various other biochemical or biosynthetic pathway. And till this point, till today, we have seen, the three important steps of cellular respiration: the glycolysis, the link reaction, the Kreb cycle. Now in tomorrow's class, we are going to learn about the electron transport chain and how is it helping in the formation of ATP molecules. Okay, that means all the NADH2 and FADH2 which are produced till date, okay, which are produced till now, we need to see how will they deliver their energy to the electron transport chain and how will the ATP formation take place. Okay guys, so that's it for today. So before taking your leave, let me again tell you my timing. I'm live daily at 5 p.m. on your favorite channel, Let's Crack NEET UG in English. Okay, and tomorrow I'm going to discuss about the electron transport chain. So this is really an important class. Do not miss it. Apart from that, daily at 9 p.m. I'm live for your practice sessions. And today's topic of practice session is special because today we are going to conduct a practice session from all the chapters of plant physiology. Okay, so I've included questions from all the chapters of plant physiology. Okay, so it will be fun to learn and to see what do we exactly know and what do we recall. Okay, apart from that, I'm also live at 11:30 a.m. on Unacademy Learning app for my special classes. Okay, well, if you have still not pressed the like button, do press the like button and share this video to the maximum, because by your sharing, maximum students will be getting benefit of this knowledge which we are continuously sharing on this platform. Okay guys, and if you have still not bought Unacademy Plus or Unacademy Iconic subscription and you're planning to crack NEET UG examination, okay, so to be under the best available guidance from under the topmost team of educators, go for Unacademy subscription, Unacademy Plus or Iconic. And don't forget to use my referral code AG001 to get an instant 10% discount. Okay guys, so that's it for today. We'll be meeting again tonight at 9 p.m. with our practice session.